Sometimes we want to take a substance and move it from its area of low concentration to its area of high concentration. Now I want you to compare this to what we've spoken about before. If something is at its high level of concentration and we want it to move down to its low level of concentration, we know this costs no energy, right? If we use the analogy of the slide, they just let go and that's what they want to do. No energy. But what we're referring to here is sometimes a substance wants to go from its low level of concentration to its high level of concentration. And if this is the case, well, the question is, how does it work? Well, firstly, if we use the analogy of the slide, they need to go from low to high by climbing that ladder. Now, climbing the ladder costs energy. And the energy, biologically speaking, is something called ATP, which is adenosine triphosphate. So if you want to go from a low concentration to a high concentration, you must use ATP. Let's take a look at a specific example. So here we have some ions sitting inside of the cell. So you can see these ions here in the intracellular fluid, and you can see the ions outside the cell in the extracellular fluid. Now, a couple of things. You can see that inside the cell in the ICF, that's the low concentration gradient, or low concentration. Outside is where we have the high concentration. Now, without energy, what this ion would want to do is actually go from outside into the cell. That's what it would want to do. But the thing is, it can't do that because at the moment it wants to go in. So how does this process work? Well, let's take a look. Using ATP, this ion will move from its low concentration gradient to its high concentration gradient. And because it uses energy, we call this process active transport. Specifically, this is called primary active transport because it directly uses ATP to go from low to high. But sometimes you can piggyback on the energy used through another process and not use energy directly through ATP. What am I referring to here? Okay, what I've got here is an example that we've spoken about previously where a substance will move from its high concentration gradient down to its low concentration gradient. Remember, this is the process of diffusion, and this costs no energy at all. But in this process of going from high to low, another substance can piggyback on the energy that this process creates. Going from high to low is great. It's what it wants to do, and something can just jump on the back of it. So what's this thing that can jump on the back? Well, what if we had some Justin Biebers here? And you can see that inside the cell, you've got a high concentration of Bieber, and on the outside, you've got a low concentration of Bieber. So to go from outside in would actually be going from low to high. And that's going to cost energy. But there's no ATP available here. So how's it going to do it? Well, it's going to piggyback on me moving in. This process is still active transport, but because it's not using ATP directly, it's called secondary active transport. And it can go in either direction. So when it comes to active transport, you can have primary active transport or you can have secondary active transport. Specific examples, because obviously there's not my head and Bieber's head floating around our bodies. We're going to have very specific charged atoms or elements floating around, right? So these ions include sodium and potassium and chloride and calcium, for example. And I want to give you an example of probably the most important primary active transport process in the body. Every single one of our 30 trillion cells does this primary active transport. Let's take a look. So here's a pump. What this pump does is it takes 3 sodium ions and throws them outside the cell. And it exchanges them for 2 potassium ions. And it does this using ATP. Now, what does this do? By throwing 3 sodium outside and 2 potassium inside, you create a concentration gradient where all the sodium is sitting in the extracellular fluid. And you create a concentration gradient where all the potassium is sitting in the intracellular fluid. So that's the first thing. It creates a concentration gradient of a chemical, but it also creates a slight electrical gradient. Have a look. The sodium each contains 1 positive charge. So you've got 3 positive charges outside. It throws 2 potassium in, each of which contain a positive charge. So you've got 2 positive charges inside, which means if you were to compare the outside of the membrane to the inside, the outside is slightly more positive compared to the inside. But let's take a look at something else that's happening here. You actually have another little channel here. And this channel is a leaky channel. The door of this channel is left open a little bit. And what this channel is for is specifically for potassium. Now, potassium, we just said, is mostly inside the cell. So diffusion occurs and potassium leaks outside, carrying that positive charge with it. What that means is the outside becomes even more positive compared to the inside. So now we've created an electrical chemical gradient, all because of this pump. This pump is called the sodium potassium ATPase pump. Really important. Throwing 3 sodium out, 2 potassium in, and then some of that potassium leaks out, making it really positive outside and slightly negative inside. Why is this important? It's really important because of many reasons, but I'm going to show you one. What we've got here is a neuron, right? This is a neuron, this orangey pink thing that we've got here. What we've got with a neuron is that one end is going to be talking to the brain, and the other end is going to be talking to muscles. And so the end that's talking to the brain says, hey, I want to dance. I want to move these muscles. I want to contract and dance. How do we do it? Well, a signal needs to be sent from point A here down to point B here. Everyone talks about the electrical signal that neurons send, but to be specific, they're electrical chemical signals. Let's take a look. What you can see all the way down what we call the axon of the neuron are a couple of channels, so you can see those channels, and you can see that we've got that positive charge outside and that negative charge inside. Why? Remember, we threw all that positive sodium out and that positive potassium in, and then that potassium leaks out a little bit. All right, so you can see all the positive sodium outside. Now what happens is, when this neuron goes, all right, I want to fire off and tell the muscles to contract so I can dance, it begins at point A. And point A will open up the very first sodium channel. What then happens is sodium will move down its concentration gradient. Sodium will move into the neuron. Now, sodium carries that positive charge with it. So what do you think that means? It means it makes this area of the neuron slightly positive inside. And this charge is the key to open up the channel for the next for the next sodium channel. So this channel that we've just opened up, this channel right here, it needs a key to open up. So you could say it's gated. And the key to open it up is a charge, what we call voltage. So we actually call this a voltage gated sodium channel. All right. So you can see that some channels actually need a key, and sometimes the key is a voltage, a charge. Sometimes the key is a chemical, and we call it a ligand or chemically gated channel. Anyway, this channel's opened up because it's become positive inside the cell, which means sodium can now move in, down its concentration gradient. What do you think that means? Sodium carries that positive charge, makes it positive inside the neuron. Opening up the next voltage-gated sodium channel, sodium moves in, makes it positive, opens up the next channel, sodium moves in, makes it positive, opens up the next channel, sodium moves in, makes it positive, opens up the next channel. What is happening? We've just, through a domino-like of nature, thrown sodium into a neuron down the axon. We've now reached the end of the axon. And this signal that we've just sent goes to the muscles and says, let's dance. This is one of the important reasons to understand both diffusion, because sodium diffused in, but also active transport for sodium-potassium pumps, because it resets neurons so that we can send signals. Now, in summary, you've got passive diffusion, going from a high concentration to a low concentration. You've got facilitated diffusion, in which this process must happen through a channel. So either the substance is too large or it's charged. And you have osmosis, which is the movement of water through a semi-permeable membrane from an area of low solute concentration to an area of high solute concentration. Remember, those solutes are pulling water towards it. All of these are forms of passive transport. You've got primary active transport going from a low to a high concentration gradient, so it's the opposite to passive, but we must use energy. If we use energy directly, it's called primary active. Or we can use energy indirectly by piggybacking on the diffusion of a substance, like what's happening here, and that's called secondary active transport. Finally, sometimes we need to bring in really big substances like bacteria into a cell to kill it off. This process, where bacteria or other big substances get engulfed by a cell, and you can see it sort of pinches itself off to form what looks like a mini cell inside, this is the process of endocytosis. Endo meaning inside, cyto meaning cell, osis is a process. So bringing something into the cell. If you're bringing in substances that are solid substances, we call that phagocytosis because it means to eat, right? If you're bringing in liquids, it's called pinocytosis because that means to drink. So phagocytosis, ingesting or eating, pinocytosis, to drink.